Hello, my name is Dr. Kristen Smith. I'm a PhD scientist that was trained at Duke University in Durham, North Carolina. I'm also a certified clinical microbiologist as a diplomat of the American Board of Medical Microbiology. Today, I'm going to be talking to you about the tools in clinical microbiology as a guide to surviving the fungal apocalypse. I will mention that these views are my own and not that of my employer. I also want to say that there may be some spoilers associated with HBO's The Last of Us show. So if you have not seen that yet, please be aware that there may be some slight spoilers. You are urgently needed in the laboratory. There is a fungal pandemic occurring, and we need you to help with diagnosis of our patients. You might have heard of the cordyceps infection. It causes coughing, slurred speech, muscle spasms, and mood changes. So we need to prepare laboratorians for these fungal outbreaks to help with us in the laboratory. Okay, this may not be accurate in today's world. This is the basis of HBO's The Last of Us, but there is a lot of talk in the media today about The Last of Us, and it's asking the question, how likely is a fungal apocalypse? Is the organism that is presented in the show able to cause human infections to the degree of what is observed in the show, creating zombies? Or is this just Hollywood doing its thing? But it does bring up some great points about a lack of knowledge in clinical mycology. So today we're going to discuss those, because the fact of the matter is clinical mycology is in need of trained laboratorians, but it's not the only clinical laboratory science that is. Most of you have already probably felt the shortage that I'm presenting over on the left. So this is a workforce projection, and what you can see is the demand far and away exceeds the number of trained clinical laboratory scientists that we have available in the workforce. So today we're going to introduce clinical mycology in case you're ever needed to join the clinical mycology laboratory at your institution. We're going to highlight some common fungal infections. And we're also going to cover the diagnostic tools that you might need to use in order to diagnose these in your laboratory. And we're also going to talk just a little bit about antifungals, the drugs that we use to treat these infections. And along the way, we're going to discuss The Last of Us, and we're going to be separating some fact and fiction so that you can take this back to your friends and family whenever they're worried about a potential fungal apocalypse, and you can let them know the real use knowledge of clinical mycology to help them separate fact from fiction as well. So next, we're going to talk about clinical mycology and the diagnosis of these fungal pathogens. But first, let's talk a little bit about microbiology and what subspecialty fields are included. I'm sure most of you are already familiar with virology, bacteriology, and mycology. I'm not showing parasitology or serology, but those oftentimes are also included in the microbiology space depending on the institution. When we talk about virology, we're talking about non-living organisms that hijack our host immune machinery, our host cell machinery, in order to create their own proteins that they need to carry out their life cycle. They are acellular. As I mentioned, they need to infect a host cell in order to survive. They're very small, 20 nanometers, and that's not something we can see with our eyes. It's not something that we can see with a light microscope, but we would need an electron microscope in order to visualize these organisms. They have relatively small genome sizes, as you can imagine. They're not making their own proteins. They're utilizing host cell machinery. So their genome sizes range from about 2,000 to 2 megabases. And moving on to a slightly bigger set of organisms in size, that is, we're talking about bacteria. These are now living. They can carry on their life cycles inside or outside of host cells, depending on the organism. They're unicellular. So everything that they need to carry out their life cycle is within their own cells. The sizes are about 1,000-fold bigger than what we observed with the viruses. They're 0.2 micrometers to 2 by about 8 micrometers, and their genome sizes are bigger as well. But let's talk about mycology. What do the organisms look like in this subspecies or subspecialty of microbiology? They're also living, but they can be unicellular, or they can form communities of living organisms and be multicellular as well. And their genome sizes reflect that. it reflects the complexity. So we're looking at about 9 to 177 megabases when we're talking about the genome. So very, very large. Dig into that a little bit further. So what is mycology? It's the study of fungal species, and there are a ton of them all over the world. It's estimated that there are about 1.5 million fungal species. Not all of them are studied or well understood. This is just our best guess. So they're everywhere. They're also life-giving. So what I'm showing up here on the top left is penicillium. So as you can guess, this is where penicillin was first isolated from. So fungi tend to live in the environment and in the soil, and they have to compete for nutrients with things like bacteria. So it makes sense that they produce proteins and other materials that they use to kill the bacterial cells around them and use their nutrients. But in this case, penicillin is life-giving and has been saving lives since about the 1940s. But it can also be deadly. So I am actually showing you the cordyceps bacteria, or I'm sorry, the cordyceps mold that is infecting a species of ant. And it does create that zombie-like facade that we see in The Last of Us. The ant acts very uncharacteristically until the mold is big enough, has taken up enough of that host cell to actually come out of the cell and cause this mycelium, the structure that you're seeing here, which inevitably leads to the death of the ant. They're also quite tasty. I'm sure some of you really enjoy mushrooms, whether it's in soup or in roasts or in a variety of different ways, but mushrooms can taste delicious. They're also very challenging to study. So I mentioned on the previous slide that they can be quite complex because they can exist as unicellular forms or in multicellular forms. Their genomes can be very big, and they're just highly prevalent, but also slowly growing. All of these factors make them very challenging to study. Let's dig into a little bit further on what is mycology. I'm sure you've heard the term yeast. So what do I mean when we talk about yeast cells? Well, these are eukaryotic cells, like human cells. They have a true nucleus, but their cell wall is made up of chitin. They are single cellular organisms, and the way that they reproduce is by budding. So if you will look down here, these are yeast cells. This is probably, a little greater than 1,000x represented. And what you can see is a lot of fungal cells. And you can also note these colorations on them. Looks like little dots on the yeast cells. And those are actually called bud scars. These are where the membranes come out of the mother cell. All of the cellular materials, proteins, genetic material get moved into the daughter cell after being replicated or increased in number. And then once there's enough for that organism to survive, it pops off. And where the daughter cell popped off of the mother cell, you're going to see this bud scar. All of you are probably very familiar with agar media or solid media or nutrient agar. It has a variety of different names. But over here, I'm showing you what a typical yeast looks like on one of these agar plates. So they're very creamy. They can be described as glabrous. Most often, they are this off-white color. Now, let's talk about molds. Two different spellings, as you'll note. The US spelling is a little bit different than the UK spelling that adds that extra U in the middle, both pronounced as molds. It is also a eukaryotic cell that has a chitin cell wall, but now these are multicellular, and they reproduce a little bit differently. They do produce these spores that help them carry out their life cycle, which are very useful in identification that we'll talk about on a later slide. When we talk about the size differences between the yeast cells and the mold cells, it's about a 10-fold difference in the screen that you're seeing here. So on the left, it's about 1,000-fold or 1,000x, but over on the right, it's about 100x. So these are mycelial cells. You can see there's a little bit darker perpendicular lines that are intersecting the cells. Those are called septate hyphae. And you can also see the spores that the organism was producing right here. What does that look like on a plate? Well, I'm also showing you that example over here. There's no less than probably 5 to 10 mold species represented on that plate. And as you can note, they are quite distinct from the mold colonies that we see on the left-hand side. So a lot larger in size. They oftentimes look fuzzy, if you will. So very different in appearance. There are some organisms that can exist in different states, in both states. And these are called dimorphic fungi, or dimorphs for short. They can exist as yeast cells when they're at 37 degrees or infecting a human host, but as a mold when they're present out in the environment. So in general, things are one or the other, but there are some specialized organisms, pathogens, that are both, that can do both. Location, location, location. Location matters. They are part of our human microbiome. And what you can see over on the right in the text, if the text is larger, that means it's present in higher quantities. And so there's some organisms that are always pretty much in our environment that we're breathing in. So Aspergillus species, Alternaria, Cladosporium, very, very common. Things that we eat, Saccharomyces, so bread. I know you know that smell when you are baking fresh bread, and that's a yeasty smell. And so it's quite tasty, also very good and nutritious for us. But because of that fungal cells that are in what we eat, it's going to be present in our intestine as well. So you can see Saccharomyces is very present, as well as Candida. There's many organisms that are also commonly on our skin, in our mouth. We're constantly interacting with our environment, and it's represented on our human microbiome. There's also a lot of variability when we're talking about geography or climate. So the human microbiomes are going to look different in individuals that live near the coast versus living in the mountains, or living towards the poles versus the equator, just because the organisms that are typically in those environments are impacted by temperature and humidity. Our hygiene, so how often we shower and bathe to mix up, if you will, the human microbiome, is impactful, as well as what we are eating and consuming. Location matters when we're talking about the type of organisms that you might be exposed to, and therefore more likely to get an infection with. So take a look at our shower. And what happens when you get in a shower maybe that looks like this without your shower flip-flops on? Well, you might be more susceptible to a fungal infection on your feet because they're touching that floor that is contaminated with the mold. What about rose gardeners? This is a really interesting one. So we can see an individual in the garden minding her own business, but little does she know that Sporothrix is present on these rose bushes. And what happens with roses? They have thorns, and sometimes they will prick us. And what you can see here are instances where this individual is probably scratched or her skin was broken by a thorn, and that allowed entry of the mold Sporothrix in there, and then that subsequently causes these skin lesions. And our jobs, where we work, can also really impact what organisms we might be exposed to and therefore susceptible to. So individuals that maybe mine or go spelunking, or farmers, what they're doing is, as they're walking and going through their work, they're kicking up dust that might be coated with fungal spores. And then because they kick up the dust, they're then breathing in the dust that's contaminated with the spores. Therefore, individuals in these careers or these hobbies such as spelunking might be more susceptible to these type of mold infections. We're going to talk a little bit more about location, and we're looking at the United States and some dimorphic fungi that are endemic here. So the disease states that we're looking here is coccidiomycosis, histoplasmosis, and blastomycosis. So cocci, as it's known as in short, tends to like arid environments, very dry and hot. Whereas histo and blasto are like more temperate climates. They also prefer more humid environments. They used to be more prevalent in the Ohio River Valley, but their range is really increasing as the temperatures are rising. So you do see this growth towards the eastern half of the United States. They overlap quite well in this image, so it may be hard to distinguish histoplasma from blasto, but they're very much covering the eastern United States, eastern half of the United States. And these can cause severe respiratory illnesses. Blasto might also cause some skin lesions that don't look much different than what we observed with Sporothrix if an individual has exposed skin on their body and come into contact with the mold. And just a reminder, the mold form will be present in the environment, but once the organisms enter our body at 37 degrees, they switch into their yeast form. And the reason for that is it's a lot easier to hide out and get around the human body when you're a smaller yeast cell compared to that larger mold cell. But the number one top fungal pathogens to be aware of are these dermatophytes. And these infect human hair, skin, and nails. They can be quite contagious, so direct contact with someone else that has the infection is enough to transmit the infection to a new host, as well as fomite transmission. So the shower would have been an example of a fomite. The organisms that most commonly cause these infections are trichosporin, microsporum, and epidermophyton. We typically refer to these as tinea plus whatever location that they're found in. So tinea capitis, that would be an infection of the hair, or tinea barbae, that would be an infection of the beard hair. But they have more common names. So ringworm, athlete's foot, jock itch. This is a good example of onychomycosis, or infection of the nail bed. That one's a little bit different in that it doesn't follow the tinea format. It has a different name. But there are also a lot of fungal pathogens that can cause vaginal candidiasis, or infection of the vagina, invasive candidiasis. So candida, if it enters in our bloodstream, can cause very serious fungal infections in our blood. And then more commonly known as thrush, these are candida infections of the mouth, throat, and esophagus. What is the economic burden of these fungal pathogens? These are estimates from the CDC, and we're going to focus on the data from 2019 over here on the right. But when we talk about those hair, skin, and nail infections, those dermatophytes, those are responsible for almost $1 billion annually when you consider direct medical costs. And we're well over $1 billion and closer to almost $1.5 billion when you're looking at the indirect costs factored in as well. So very high economic burden. When we look at just Candida, we're adding on an additional $4 to $5 billion in costs due to Candida infections. And then some of the other organisms that we talked about, such as those dimorphs, when you consider all of these fungal pathogens and their direct cost estimates in 2019, we're looking at over $12 billion. And then these are just the cases that we know of. So now let's switch gears. We talked a little bit about the pathogens, but let's talk about how, if you were in the clinical mycology lab, how would you go about identifying them? You can do it direct by eye. So we're going to talk a lot more about this solid media. It works for some molds, but not all. So on the top left, this is Aspergillus niger. As you can see, it has very dark black coloration on a plate. It also looks a little bit sandy or granular. So when you see that identification, and you can automatically say, that's very suspect for Aspergillus niger, and give out an identification. I will caution that the plate type matters. So there's a lot of different types of nutrients in a variety of plates. And that can impact what the mold looks like when it's growing on that solid media. So it's important that you understand what the mold looks like when you're plating it onto a certain media type. Detailed descriptions are very necessary. Does it look cottony or granular like we're showing here? What color is it? It helps if you don't say red, but maybe it's a bright maroon color. So as more detail as you can provide in the description, the better. Now I'm going to channel Missy Elliott. Is it worth it? Let me work it. Put your plate down, flip it, and reverse it. Why is that important? Molds look different on the top and on the bottom. So down here on the left, this is T. rubrum, or Trichophyton rubrum, one of our dermatophytes. It looks very white and woolly when you're looking at it on the top. But when you flip that plate over, it has a variety of colors. So brownish to maroon colorations that are have gradations all the way to maybe an orange and yellow. So it's very distinct for this organism, and a lot of trained mycologists can get this identification just by looking at a plate for both of these organisms. But I will say training clinical mycologists for identification by eye, by both the colony morphology and on the microscopic morphology that we're going to see on the next time, is very labor intensive. And in order to be very, very well trained, it takes 6 months to sometimes upward of 2 years before your clinical mycologists feel very comfortable in this particular area. Obviously, you're looking for growth on a plate, so that organism growth is required. So we're going to call back to The Last of Us. Can we identify the mold just by looking at it? Well, if you're familiar with the show, then you know that this is what an infected looks like. And I think you can say, yeah, this is in the show, the cordyceps bacteria. So maybe this is a fact, right? You can identify it by eye. We're going to talk about microscopic examination. And what does that look like? Well, you're going to take a mold and you're going to take a slide and you're going to take a stain that helps you visualize the sporulating structures that you're going to use for identification. You're going to look at that slide through a microscope and hopefully get your identification. But sporulation is needed, or else it's just going to look like a bunch of hyphae that have no discernible structures on them. So again, we're going to go back to our plate. This is Aspergillus fumigatus. This is what it looks like on a plate, a very beautiful green color that kind of gradates all the way out to the end. We have a couple different methods. We have the tape prep method, and it's pretty much what you would expect. You take a piece of tape, you fold it over, and you touch the top of that mold colony very gently, and then pull it back, and then lay it over some stain on a slide. This is all done in a biosafety hood or a BSC, because these create spores. So fungal spores will move out into the environment if they're disturbed at all. And so that happens when we perform a tape prep, and we want to do that where we're not at risk of breathing in those spores. So you push the tape down on the slide over that stain that's going to help you visualize and to kill the organism that's present, and then we can go to the microscope. Alternatively, in particular, when we talk about dimorphs, these are very virulent. You want to minimize the risk of transmitting these infections as best as you can. So you don't want to create a bunch of spore release when you pull off that Scotch tape from your mold colony. So it's better to use your utensils, metal utensils that are similar to dental tools. You pick a colony off of that plate, then you put it inside of the stain that is on your slide, and then you gently tease it apart. And that's called the tease prep method. After you put a cover slip on, you're also ready to go to your microscope and look at some structures. So this is what Aspergillus fumigatus looks like under the microscope. This is at 100x. This is also Aspergillus fumigatus, as are these two. So I'm showing 4 different examples of the same organism to reiterate the fact that identifying mold by microscopy is very, very tough. And it takes a lot of experience for an individual to be able to say, yes, this is the same organism as this and this and this, and they're all the same. This is just one example. There are hundreds of molds that may be identified by microscopic methods, and some of them look very similar to one another. So it's important to have a lot of experience in this method. You don't have to just memorize these. There are reference materials available. So you can look at your microscope, take your reference material, and ask the question, does this structure look like this organism, to make your identification. There are other colorimetric assays that use biochemistry, and they really play on the fact that organisms are different from one another, just like you and I are different. Some of you might be lactose intolerant, whereas I love eating ice cream and would be very sad without it. There are organisms that are very specific in their nutrient requirements, so they may or may not grow in certain conditions. We can utilize biochemistry that if a certain sugar, for instance, is utilized by the organism, it shows a color change. And by using that technology, we're able to discern organisms just by visually looking at color changes, colorimetric assays, if you will. So we have this technology in solid media. This is a plate that can distinguish different species of Candida. This is another tool of using liquid media this time to look for growth that causes color changes. And this is yet again another solid media type that will exhibit a very prominent color change if this nutrient is being utilized. I think it's very important to note here as well that growth is needed. But what if we don't have growth? Well, then we can utilize ID by antigens or antibodies. This is generally used with clinical specimens such as blood and urine and CSF and BAL. And we can detect the organism's antigen. So that's that component that the organism is making that our host cells recognize as not human, not us, and will cause a response if they're present in the human body in general. We can utilize ELISA methodology to detect and quantitate the antigen present in those specimens. We can also use lateral flow assays, which we're all familiar now after COVID testing. I'm sure we've all given ourselves COVID lateral flow assays, so you're familiar with the concept of one line equals your control work, and two lines means that your antigen is detected. And so there are a lot of different antigen and also antibody tests that are very similar to what I just described for fungal organisms, and those include the Aspergillus, that includes the dimorphic fungi, Coccidioides, Histoplasma, and Blastomyces, as well as Candida and Cryptococcus species, as well as looking for beta-D-glucan, which is present in fungal membranes. Sometimes these tests have problems with sensitivity and specificity, so if these are being employed, it's important to know the limitations of your assay. In particular, the dimorphs tend to cross-react with one another, so again, it's always important to know the limitations of the assays that you're working with. But it does have the added benefit of being culture independent. We can also use molecular assays for identification. We can use the detection of proteins in the form of MALDI-TOF, or matrix-assisted laser desorption ionization time of flight. Say that 5 times really fast. But you do need organism growth. And the basic concept of this is that you take an organism, you shoot it with a laser, and you quantitate those protein fragments in a way that will give you an identification. A cartoon showing that is we have our sample of organism growth. We'll put it on a slide generally, put a matrix solution over top of that, overlaying that to allow for the proteins to be ionized. After they're hit with the laser, they'll flow through a vacuum. The smaller charged particles, proteins, will go through that membrane, that vacuum faster and cause the peaks towards the front. And then the larger the size, the longer it takes to travel through the vacuum. And based on these patterns, using a known database of organisms, you can get a very good identification off of that protein analysis. This has the benefit of working with both yeast, the single cellular organisms, and the mold, the multicellular organisms. But you are only as good as your database. So if an organism is not present in your database, you will not get a good identification. The gold standard can be thought of as our nucleic acid detection tools. There are now syndromic tests with a limited number of organisms. So by utilizing PCR to amplify sections of these organisms' genomes, you can quickly know if one of a panel of organisms is present in direct from your sample. You don't need organism growth for this. You can also use sequencing. So you can use a Sanger-based methodology and look at the ITS gene for identifications, or you can utilize whole genome sequencing if that is available at your institution. And you're going to get a nucleic acid chromatogram that looks something like this. You can compare that again to a known database to get your identification. And the added benefit of these assays, they can come direct from specimen, or you can utilize your cultured organism to get your identification as well. So going back to The Last of Us, they have this wonderful tool from their Federal Disaster Response Agency, or FEDRA, that all they have to do, it looks kind of like a temporal scanner whenever they're using it in the show. And basically they have an identification of positive or negative in seconds. And I'm going to go ahead and say that is fiction. There are incidences of really fun things like dogs that are able to sniff out infections or people that are infected, or things like rats. But as far as having a tool that we can use in the clinical space to rapidly detect direct from a scan of the human, that's obviously fiction for right now. So pathogenic fungi are all around us, and they are potentially very deadly. So who is at risk for these infections? It's typically those that are immune compromised, individuals with HIV or cancer, the very, very young or the very, very old, those that have compromised skin or don't have a protective barrier that might allow for those organisms to get inside our bodies. An interesting example is HIV and Cryptococcus. So Cryptococcus species, Cryptococcus neoformans primarily, when we're talking about HIV and co-infections. This was an organism that wasn't thought to be pathogenic. However, when the HIV pandemic occurred, there was a lot of individuals that were immunocompromised and were found to be very susceptible to these infections. It causes a very serious infection and can cause lesions in the brain that you're seeing here. So individuals with HIV are unfortunately at greater risk of fungal infections. Individuals that have antibiotic treatment. So when we wipe out our normal microbiome, we're lessening the competition for the fungi that are present in our body that I showed on an earlier slide to really increase their presence and potentially cause an infection. These are called opportunistic infections. And so I'm going to show you now, this is what thrush looks like. This is a baby that potentially had, maybe an ear infection, was treated with an antibiotic, and then later on developed this white material on their tongue. And that's actually Candida that's growing on this baby's tongue, and it's commonly referred to as thrush. With COVID-19, we also saw some co-infections that were pretty prevalent. The first is Aspergillus. So all of the examples that I'm going to show you are taken from skin or from biopsies of tissue. And so this is a stain, a GMS stain of what Aspergillus looks like inside tissue. You can see, maybe not appreciate the septations that are present in these hyphae, or those little perpendicular blocks that are present in the cells. And you can also note some branching. Now, I will caution and say that some, it maybe used to be thought that if you saw 45-degree branching, which this present probably here is the best example, and have that be indicative of an Aspergillus infection. That is not true all the time. It's true a lot of the times because Aspergillus seems to cause a lot of infections in humans, but it's not good enough to see that branching 45 degrees and say you have Aspergillus alone. We have another organism called mucor. It's a little bit different in that it has no septations. So it just looks more like worm-like structures going through. They're a little bit more broad hyphae present. And then we're going to look at Candida species. So you might say, hey, Dr. Smith, you told me that Candida were yeast cells, and I'm seeing things that look a little bit like hyphae. Well, these are called pseudohyphae, and what the Candida species is doing is looking for nutrients. So it's actually growing inside the tissue, searching for nutrients that it can utilize, and is forming these kind of false hyphae in its search for nutrients that it needs to survive. Now, I think that fungal infections are probably greatly underreported. And the reason for that is because there are a lot of over-the-counter medications that are used to treat things like the dermatophyte infections, the hair, skin, and nail infections, as well as vaginal candidiasis. So these are out there. So I do think that these are underreported. And to further discuss reporting, we do not have consistent national or state reporting for fungal infections. And so if you look at blastomycosis, for instance, Arkansas, we're only going from Alabama to Idaho here, but Arkansas is the only one that has it as a state reportable. And it's a variety. No two states really look the same. I will say there is one organism present on here with the asterisk. That is Candida auris. We're going to talk a little bit more about that, but it is a nationally notifiable disease. But unless we have standardized reporting across the states and at a national level, I think we are at risk of not having a great understanding of what our case numbers of fungal infections actually are. And I hope that I've convinced throughout the talk that identification is hard. It takes a lot of training in mycology to do our identifications by eye. The organisms grow very, very slowly. So if we're looking at solid media, yeast take anywhere from maybe 2 to 7 days to be present on a solid media plate. Molds take even longer. Some of them will grow, you can get some good growth maybe in 2 days. That's very rare. On average, it's a week or more. And a lot of clinical mycology labs hold their plates for 4 weeks looking for molds. So it takes a lot of time to visualize these organisms sometimes, especially when you compare them to things like bacteria that maybe show up in 16, 18 hours, and maybe the plates are checked after 24 hours. Fungal infections are tough and challenging to treat. So we have 4 drug classes, and I'm showing you here what they are, but 3 out of the 4 of those are impacting the membranes of molds. And so there's not a lot of diversity. We have an example too of the polyenes. We have amphotericin B. It's a very effective antifungal agent. There's not a ton of resistance. However, it prevents the conversion of it's binding to sterols is its mechanism of action. And human cells have sterols as well. And so what does that mean? It means if you're getting treated with amphotericin B, some host cells are being negatively affected and dying as well. So, we have to really worry about organ failure when treating for a long time with amphotericin B. What does the drug line look like? Well, we're in the process of developing some second and third generations for our polyenes and our azoles. And there are some future generations that are currently being investigated, also to include the echidocandins. We're looking at changing the delivery approaches. So maybe using granulocytes to get these antifungal medications to the site of infection. So we're reducing toxicity up to elsewhere in the body. So that's one way of potentially improving the use of what we currently have. Or maybe repurposing other drugs, such as rifampin, to treat fungal infections, and studies are being done in that arena as well. And also looking at maybe modulating our immune system some way, similar to CAR T cell therapy. You're looking to add activated cells and use that as a method to infect or attack the yeast cells that are present in our bodies. I will also note that there's currently no vaccine available for any of these yeast or molds that I've discussed. So we can't talk about fungal infections being tough to treat if we don't bring up something that is very important and new. So Candida auris is an organism that was first identified in Japan in 2009 and has since spread globally. It has intrinsic resistance. So maybe you remember the old Maybelline commercial. Maybe she's born with it. Maybe it's Maybelline. Well, Candida auris was born with intrinsic resistance or innate resistance to 90% of the time one fungal class and 30% to two antifungal classes. So there's already a limited number of antifungal classes, and now we have this organism that's novel, that's already spread around the globe, that's resistant in 30% of the time to two of our entire classes of antifungal agents. It's very scary. What's also scary is that there is a very high mortality associated with Candida auris. Depending on the case studies that you can review in the literature, that mortality can be close to 70% at the high end of that scale. So I would say on average, mortality is in the 30 to 40% range, but that's a very high number. We tend to see these infections in people that are already at risk. So a lot of cases in long-term care facilities and in the elderly, or people that have underlying conditions such as diabetes mellitus. There are also disinfection challenges that have led to outbreaks because this is a very hardy organism. The disinfectants that are commonly used in places like long-term care facilities are actually not adequate to kill the fungal spores of Candida auris. So you think you're cleaning your room space really well, but that organism is actually resistant to those cleaning methods, and it is able to grow and be transmitted if someone touches that infected surface, for instance. So where have we detected cases in the United States? This is a heat map, and this is of all cases that have been reported to the CDC. So the darker the red, the more cases there are. And so you can see that we have had we've had outbreaks in New York, Illinois, and California, but really it's only a matter of time before we see this in every state. These microbes, they don't know boundaries of state lines. They don't know, one country versus another. They're going to transmit as their host does. And what do the cases look like from a numerical scale? So we're only showing, remember, the first case in the United States was in 2016. It became nationally notifiable in the United States in 2018. So you can see that the case numbers weren't really growing and maybe even decreasing, because what happened in 2020? COVID-19 pandemic came. No one was paying attention to Candida auris anymore, wasn't being tested for, wasn't being screened for. But when we started looking again, you can see that the number of cases had sharply risen. And after we noticed that, there was a lot more of these in the lighter blue, these screening tests that were being performed. And the more you screen for something, the more you're going to see. And so that's what we're seeing in this graph. But we are now up to the level of seeing 150 cases of these every single month in the United States. So it is growing, and we need to recognize this novel pathogen. And I'm going to stop for a second because there are spoilers on the next slide for The Last of Us. So if you do not want to be spoiled at all, this would be a good time to stop enjoying the presentation. But now we're going to talk about The Last of Us, fact or fiction. So in the show, they had transmission through these mycelial cells that came through the mouth, so transmitted orally and through bites. But I hope that I've convinced you that that's not the typical route that yeast and molds take to cause infections. It happens most often by touching an infected surface or interacting with someone that might have an infection, or breathing it into your lungs. Those are the two primary methods of being infected with a yeast or mold, or undergoing antibiotic treatment or being immune compromised and letting the natural flora let opportunistic infections of our natural fungal species rise up and cause infection in us in an opportunistic way. So I'm going to go ahead and call that fiction. Treatment. So in The Last of Us, there was no treatment for their cordyceps infection. So individuals had no chance. They were going to turn into an infected within, depending on the type of exposure that they had, 12 to 48 hours. Well, I'm going to put that as a question mark. So we just talked about Candida auris, and it's concerning their intrinsic resistance to our classes of antifungals. And so while it's not fact right now, it's not so far removed of a possibility that we could potentially see a completely resistant fungi at some point. The timeline. So in The Last of Us, it went from a normal day to the end of the world in, I believe, a weekend. So Friday to Sunday, the world had ceased to exist as we know it. The good thing is, I think that this is fiction. So we talked a little bit about the growth rate of yeast and molds, and they are a little bit they are slower growing than things like bacteria, and they don't transmit as readily as viruses. So I think that 3-day time period is really fiction. Candida auris is a good example of that novel pathogen spreading globally, but that took from 2008, 2009, all the way to 2016 just to get in the United States. There have not been cases in every single country in the world. So that is at least good news. But global reach. Just talked about Candida going around the world. So yeah, I do think that fungi do have very good ways of global reach. As we move, organisms are going to move. Natural immunity. So in the show, one of the main characters does have immunity to this infection, and there are individuals that seem to be more resilient against antifungals or fungal infections than others. So yes, that's probably a fact. As well as the big question of the day is there an organism out there that can cause fungal zombies in humans? I think the likelihood of this happening is very, very low. Yeast and molds tend to have a very narrow window of where they like to grow and be happy, and the likelihood of those organisms also being able to cause a human infection as they're being, attacked by our host immune system is probably pretty low. And so I think that while ants may, have some problems with cordyceps bacteria or cordyceps fungi, I don't think we as humans have to worry about turning into fungal zombies at this moment. And so with that, I thank you for your time and attention. I hope you had a really great introduction to clinical mycology. Thank you very much.